Hello, my name is Yihong, and today I'm going to introduce you about microplastic pollution in deep sea sediments and organisms of the Western Pacific Ocean. So, to speak of the microplastic, first we have to know the amount of the plastic gets into the ocean. And in the previous studies, we have known that the annual plastic production worldwide is about 350 million tons per year in 2018, and 10% of them will eventually get into the ocean, and the sizes are ranging from meters to nanometers. But in this study, we only choose those who are under 5 millimeters to be microplastics. So, how microplastic can affect the organisms? First, it will increase the energy expenditure. Means you have to use more energy to eat those microplastics and throw them out of your body, or it will cause physical blockage. That means the microplastic might stuck in your body, or cause full satiety. Satiety means if you eat something and you feel full. The feeling of full is the satiety. And then those microplastics might have some harmful additives, and it will release if you eat them in your body, like pesticides. And those are mostly harmful or toxic. And the microplastic will absorb some persistent organic pollutants called POPs or heavy metals, and both of them are toxic. And then I will introduce you about the PCBs. It's another subject in our study. And this PCBs is a kind of substance with strong toxic, and it can cause cancer and neurotoxicity. And it's a kind of POPs, and that means it can stay in the environment for a very long time. So this is our research purpose, and we want to understand the distribution and the abundance of microplastic and PCBs in Western Pacific deep sea area, and to more comprehensively understand the correlation, means the relationship between the microplastic and PCBs. And we want to investigate the microplastic contamination of deep sea organisms. So this is our sample size here. As you can see, Japan is here, and China, we are here, and we'll talk about it later. So how do we get our samples? This is the sediment samples. We use the box core to get the sample, and we will retain 0 to 5 centimeters of surface sediments in glass bottle because we want to prevent some possible plastic contamination. And for biological samples, we use remotely operated vehicles, the ROVs, equipped with mechanical arm here and a suction hose here. And this tube will suck the biological sample to keep the samples to be complete so we can use it for analysis. So these pictures are the biological samples we get. We will classify them into orders because we don't really know what their species are. We just need to know whether they have microplastic inside them or not. So for sediment samples, we separate them into 2 portions for analyze. And the microplastic analyze and the PCBs analyze. And we'll only talk about the microplastic analyze here. So first, how to separate microplastic? First, we will add saturated sodium chloride solution, and then we centrifuge them, and we'll collect the supernatant means the liquid up here, and after the centrifuge, the microplastic will flow here. And then we filter the liquid up here to get the microplastic. After we get the microplastic, we will use the microscope to characterize their shape, their color, and their size. And then we use the micro FTIR, the machine here, to know their polymer types. And how do we use how the micro FTIR works? First, we will scan the sample with infrared light and get a spectra. Infrared light is a kind of invisible light, and this is a photo taken by infrared. And the spectra looks like this. We use the different wave numbers of light and see how many light can get through our sample to get its transmittance. And eventually we'll get a graph like this. And then we can plan the spectra with the polymer library to know what this graph represents, means what our samples are made of. So in our result, we get the average of 240 items per kilogram dry weight sediments in our sampling area. And the highest one here is 1,042 items. And in this area, we found that there are really high abundance of microplastic. And in our sampling area, we also find that we have more average items than other deep sea area like Arctic. And it's more likely to some coastal area in Malaysia or Bay Area in Thailand. And this is because our sampling area here is really close to the western garbage patch. And in the sea surface, there are really high concentration of plastic that over 33,000 items per square kilometer. So in those microplastics, we know that their colors are mostly blue, their shapes are mostly fiber, and their sizes are mostly around 100 to 500 micrometers, and their polymer types are mostly PE or PET. So in the concentration of PCBs here, let's compare it with the abundance of microplastic. And then we found that in this area has the most high concentration of PCBs and the abundance of microplastics. And in statistics, we found that there are really high correlation between the PCBs and the microplastics. So we can check the first and the second purpose in our study. We found that in our sampling area, there are higher number than other deep sea area, higher number of microplastics than other deep sea area. And then there are high correlation between the microplastic and PCBs. And in biological samples, we found that their microplastic shapes are of fiber, and their sizes are ranging from 100 to 700 micrometers, and there are 0 to 3 items per sample. So we know that the microplastic really enters the deep sea organisms. So we can check the third one. We know that the microplastic will be indeed ingested by organisms. So in the conclusion, this is our findings. We found that the microplastic invaded the deep sea and will be ingested by organisms. And then we know the amount, their shape, their color, and polymer types of microplastic in our sampling area. And we know that there are high correlation with microplastic and PCBs. And there are different abundance of microplastic with other deep sea area than our sampling area. So for future study. In our study, we have high probability of false microplastic, but it can be solved with just need to dye the sample with fluorescent dye. And this kind of dye can make the plastic to be more obvious. And we need to predict the potential risk of microplastic pollution in deep sea. And this will need to know the sinking mechanism and toxicological effects. Means we need to know how the microplastic here will influence the organisms in deep sea or affect the deep sea environment. Thank you for listening.
I think Ito makes a good conclusion, and also he explained the difficult words by pictures and explained it clearly. I think it is really great. And there is still some space for you to improve is that maybe next time you can explain the site's distribution clearly.
I think that you guys you explain your figures very well, and the slides were very easy to read. But I think in some pronouns has to be improved.
For
example, like I don't really remember, but some difficult words was pronounced very.
I think you gave a good talk, but some parts in the figure and charts are not explained. Some items in the picture.